Selamat pagi, Dokter Sulantari.
Selamat pagi, dokter.
Selamat pagi, Dokter Budi.
Dokter Budi. Wah, ini pakar-pakar berbau laut semua ini. Dokter Budi.
Sudah nggak asin,
Sudah nggak aktif.
Sudah kering, nggak asin.
Oh, sudah nggak. Kalau apa jellyfish box kan tetap aja di apa? Di air payau bisa ya? Air payau bisa ya?
Box jellyfish.
Box jellyfish itu bisa di air payau atau karena Prof. Yana Gihara pernah kita ajak ke mangrove. Mangrove itu kan mangrove itu masih asin ya? Masih asin ya? Mangrove-nya Surabaya. Oh, masih asin ya?
Asin.
Oh ya. Tapi nggak ketemu waktu itu. Hampir nyelam dia. Hampir nyelam loh Dokter Budi.
Hampir nyelam ya.
Sudah pakai baju selamnya itu. Hampir aja dia mau turun. Saya bilang tunggu. Saya takut juga kan di mangrove-nya Surabaya itu kan.
Oh, di mangrove Surabaya sini.
Pernah tahun 2019 ya Dokter Winta ya.
Iya, Bu
Saya
Dekan.
waktu itu masih melo pendekan, saya temani beliau, terus kemudian besoknya sama Dokter Winta dan Dokter Jati ditemani ke Madura, ya ke Sampang ya, ke Sampang, terus membuat sosialisasi penyuluhan, ya. Dokter Winta itu nanti kerjasamanya kan sudah diajukan ya. Dilanjut.
Iya, dilanjutkan. Beliau sudah setuju, Bu Dekan, untuk melanjutkan.
Ya.
Selamat pagi Bapak Rektor, selamat pagi Bu Dekan.
Selamat pagi.
Selamat pagi. Ya, Pak Jati, alhamdulillah kita bisa ketemu lagi. Ya, situ bisa disapa itu Mrs. Angel Yana Gihara. Ya, alhamdulillah.
Dokter Winta panggilannya beliau Prof. Angel apa Prof. Yana Gihara ya enaknya?
Kalau ada gelar depannya panggilannya nama belakang Bu Dekan, Prof. Yana Gihara. Tapi kalau tanpa gelar panggilnya nama depan.
Angel.
Iya.
Oh ya, berarti Prof.
Yana Gihara.
Yana Gihara aja.
Bapak Rektor udah.
Oh. Siap Pak Rektor.
Mohon izin Bu Dekan, ini
yuk.
9 lebih 2 menit mungkin bisa dimulai gitu. Oke. Dokter Sasti.
Ya, silakan.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Om Swastiastu, Namo Buddhaya, salam kebajikan. Yang terhormat Rektor Universitas Hang Tuah, Laksamana Muda TNI Purnawirawan, Profesor Dr. Insinyur Supartono, Magister Management Certified International Qualitative Research beserta jajarannya, yang kami hormati Laksamana Pertama TNI AL Purnawirawan Dr. Sulantari SPTHTKL, selaku Dekan Fakultas Kedokteran Universitas Hang Tuah, beserta para wakil dekan, yang kami hormati pemateri yang ada pada hari ini, yaitu Profesor Angel Anna Yana Gihara, PhD. On behalf of the organizer, I would like to extend my warm welcome to Profesor Angel Anna Yana Gihara, PhD. Serta yang kami hormati para peserta kuliah tamu sekalian. Sebelum kita melanjutkan acara kuliah tamu pada Sabtu yang berbahagia ini, marilah kita membuka acara ini dengan memanjatkan doa kepada Tuhan Yang Maha Esa, karena atas rahmat dan hidayahnya kita dapat berkumpul dalam keadaan sehat walafiat. Berdoa kami persilahkan. Berdoa kami cukupkan. Berikutnya adalah menyanyikan lagu Indonesia Raya.
Tanah airku. Kiri pandu hidupku. Kebangsaanku.
Mohon Dr. Budi di mute saja, Dr. Budi.
Berikutnya, sambutan Rektor Universitas Hang Tuah. Kepada yang terhormat Laksamana Muda TNI Purnawirawan, Profesor Dr. Insinyur Supartono, Magister Manajemen Certified International Qualitative Research, waktu dan tempat kami persilahkan.
Terima kasih. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi dan salam sejahtera. Om Swastiastu, Namo Buddhaya. It is a blessing that all of us are in the state of good health, so we can attend and expect lecture organized by the Faculty of Medicine Hang Tuah University with the Dean Dr. Sulantari. I am Professor Dr. Supartono, the Rector of Hang Tuah University. I attend this occasion with Vice Rectors first, Dr. Dian Lawarnamati, Vice Rector second, Dr. Bambang Sucahyo, and Vice Rector third, Dr. Sulistianto. It is a privilege for us to have Prof. Angel Anna Yunagihara from the University of Hawaii at Manoa, Hawaii, United States of America, as one of the experts in box jellyfish information and first aid. Aloha, Prof. Yanagihara. I am also happy that this lecture is also attended by medical student, lecturers, and doctor from Dr. Ramlan Naval Center Hospital, Dr. Tri Maharani from the Indonesian Toxinology Association, and Dr. Munawir from the Faculty of Medicine, Jember University. I also welcome around 20 graduate students from the Polytechnic University of Philippines, Laila Norma University, the University of San Carlos, and University of the Philippines Dilmans Campus. Kamusta, hello. Let me introduce Hang Tuah University to you. Hang Tuah University is a private university under NALA Foundation of the Indonesian Navy. Our vision is to become a world-class center for the development of marine human resources and marine science, technology, and arts. The vision is applied in your education, research, and community service goals. Currently, we have 6 faculties including Faculty of Medicine and 1 diploma program. We also have laboratory facilities that support our goal, include the new integrated biomolecular and hyperbaric laboratory and the Faculty of Medicine, Tuah University. I think it is a great opportunity for the Faculty of Medicine and Tuah University to have potential collaboration with the University of Hawaii at Manoa, some university in Philippines, and Jember University that share the same interest in maritime, marines, and the health of coastal community. Therefore, this special occasion of experts lecture has to be continuous with Zoom meeting to explore potential collaboration in education, research, and community service. Once again, thanks you to Prof. Yana Gihara for taking the time to give the lecture. Mahalo, Yahoo, thank you, aloha. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, salam sejahtera, Santi, Santi, Santi Om, Namo Buddhaya.
Terima kasih Bapak Rektor yang telah berkenan memberikan sambutan. Acara berikutnya adalah acara inti, yaitu kuliah tamu yang akan dibawakan oleh Prof. Yana Gihara dengan judul Box Jellyfish and Phenomenation and First Aid. Adapun sebelum kita mulai, izinkan saya menyampaikan sebagian kurikulum vitae beliau. Angel and Yana Gihara, PhD, Associate Research Professor dari Pacific Bioscience Research Center, School of Oceanography, Earth Science and Technology, Department of Tropical Medicine, John A. Burns School of Medicine, University of Hawaii, Honolulu. Pendidikan terakhir beliau yaitu PhD Cell Molecular Neuroscience, Bakersfield Laboratory of Neurobiology, Pacific Biomedical Research Center, University of Hawaii at Manoa, Honolulu, Hawaii, with dissertation a novel approach to structure function relationship of the sodium-ion channel. Beliau juga aktif dalam beberapa aktivitas antara lain ada penulisan dan juga penelitian serta beberapa kali hadir ke Indonesia. Adapun hadir pada tahun 2016 dengan penelitian bersama Universitas Jember dan juga dengan Universitas Hang Tuah pada tahun 2019. Sekian kurikulum vitae dari beliau. Now, we come into main point, that is expert lecture, Box Jellyfish and Phenomenation, and First Aid, which will be delivered by Prof. Yana Gihara. Prof. Yana Gihara, the time is yours.
Thank you very much. It's a great honor to be with you, even though it's electronically and we're going across the planet and so many time zones. I'm very happy to see all the familiar faces and I'm honored with this invitation. I will try now to do the screen share. Let's see. Is that working properly? Yes. Okay. Can everyone see the title slide?
Yes.
Okay. Clearly.
All right. Well, thank you very much for this wonderful opportunity. And I want to just check in with you. I have actually 3 lectures and I could easily present for 2 hours. Could you let me know exactly, should we take some breaks, or how would you prefer it to be arranged?
Maybe you could take the break, and some students can ask you some questions. Is that okay?
Yes. So perhaps I'll speak for about 30 to 45 minutes and then take a break and then continue. Is that a good approach?
I think that's a good approach.
Okay, good. Very good. Thank you. So the last time I spoke in person at Tuah University, I was quite happy with the level of engagement among the students. I also had the pleasure of doing a lab practical, and I found the students there to be extremely bright and engaged and very interested in this phenomenology. One challenge that I did discover, though, was that the English language presented a little bit of an obstacle to full understanding. So I'm very honored to have a taped talk, a brief talk by Dr. Al Munawir in the Indonesian language. So what I'd like to do is give a very brief introduction from a few of my slides, and then present Dr. Munawir's talk, which is all in Indonesian language, and then continue on in my more in-depth talk. And I think that way the students will be able to have multiple levels of understanding. They should be able to take away a good overview of the phenomenology in Indonesian language, and then also have the deeper details. Unfortunately, those are just in the English language, but I think for the most part, the students are quite capable. So I hope that this approach is helpful. So to begin with, basically, I think most of the attendees here are quite knowledgeable about the serious health threat that these box jellyfish present in the Indo-Pacific. The most tragic of these stings occurs in children, and many of these stings lead to death. So clearly there's a critical need for clinicians and other healthcare providers in coastal communities to have a good knowledge about these animals and how they cause such horrific injury and even death. So my career has been really focused on this for the last 24 years. I myself was stung very badly, and I'm lucky to have survived because I was over a mile from the shore as a swimmer when I was stung in Hawaii by a box jellyfish around the neck and arms and legs. This led to a horrific event where I couldn't breathe. My lungs started to fill with fluid. I was wheezing. It was very early in the morning and there was no one around me. So I was left to my own to try to open my lungs. I went to my back to try to breathe more, but basically nothing I did helped. And by the time I got to the shore, I completely passed out, and I was resuscitated in an ambulance. So I have firsthand knowledge about how serious these stings can be. But the animals that we have in Hawaii, the Hawaiian box jellyfish, is a box with the same kind of body as the larger Indo-Pacific version, but these rarely result in death, although we do have deaths in Hawaii due to this animal. Let me see, to advance. Sorry. Okay, so why does this topic matter? Essentially, as I mentioned to you, there's an incredible health burden, and presently this is an unmet need area in the Indo-Pacific, where we have folks who are being treated, sorry, technical problem here, sorry. Okay, sorry, the advance was blocked there. Okay, so understanding stings. In the event of a box jellyfish sting, the venom isn't simply on the surface of the skin. It's actually physically injected into the tissue, 1 to 2 millimeters deep. So today I will describe for you in detail what can happen after a sting. So there's local tissue damage, as well as systemic effects, including acute cardiovascular failure and insufficiency, such that at high doses, this can lead to death due to cardiovascular collapse. At lesser doses in certain box jelly animals, one can see Iroconji Syndrome, and I'll describe to you what that is. That's a slower onset disorder, but death can also occur at 4 to 48 hours after the sting. So today I will also discuss what is in these venoms and the pathophysiology. How do the various components of the venom cause these various effects? In order to really dissect the biophysical characterization of these effects of the venom, I have developed various models to understand the venom activity. And these models include hemolytic time course, as well as in vivo models of anesthetized mice and piglets with long time courses. We've worked with the US Army and have an anesthetized piglet model where we can track the animal's time course over 8 hours after the application of live tentacles. So this really provides a great insight into the pathophysiology of the venom and each of these components and what they cause. So I'm going to give you an overview of that as well today. In addition to these bioassays, I've worked to develop live tentacle bioassays in which we've taken freshly caught box jellies, mainly from the Indo-Pacific, these lethal chirodropids, and lay them on blood agar assays, blood agar plates, such as one finds in the microbiology departments of most hospitals in order to swab throats and do cultures. McConkey blood agar is a very powerful tool to look for hemolysis. Similarly, the box jellyfish tentacles release compounds which also cause hemolysis. So we've utilized this as a very powerful bioassay, and Dr. Rienta has helped perform this in the field as well. Then I will discuss additional bioassays with piglets with live tentacles. So putting all of that together, our ultimate goal is to provide evidence-based first aid and clinical management tools so that we can improve the outcomes of serious stings. Why this topic matters? Well, clearly box jellyfish envenomations are a significant and growing medical threat in the tropical and subtropical regions, particularly the Indo-Pacific. Most of the deaths and serious injuries that occur involve rapid progress of the pathophysiology in the absence of advanced medical care. So this really puts the onus on coastal communities to have prepared standard operating procedures in place before these things occur, as most children die within 2 hours of the sting and there isn't time for transport to advanced care. The injuries are often necrotic. And despite this kind of urgency, one finds contradictory and deleterious first aid. We find in going out to the fisher folk along coastal communities in Indonesia and in the Philippines that many fishermen are advising the use of gasoline as a first aid to pour over the sting. We've queried this and we've done, in fact, assays to look at the effects of gasoline, and you won't be surprised to hear that it actually was not effective at all as a first aid and was rather dangerous and caused much greater harm. So a critical barrier to progress has been the lack of understanding of basic pathophysiology and the adoption of a lot of anecdotal claims of first aid, such as gasoline. So for all of these reasons, this topic is important. And as far as digging deeper into potential mechanisms of pathophysiology, this requires a concerted long-term approach. So I'm very delighted to have the collaborative interest of the folks there in Indonesia, and especially at the Tuah School of Medicine, because this really provides us with a springboard to make positive moves forward. Okay, so at this point, I'm going to switch gears for a moment to share we'll see if this works or not, and I appreciate everybody's patience. This is a fine talk that Dr. Munawir is narrating in Indonesian language. Is that working? Or? Perhaps I'm not able to do that.
Okay.
Angel, I'm sorry if.
Okay, so perhaps
if
I'll try. Okay, let me continue then. Sorry about that. It would have been a nice idea if it worked, but I don't know the technical part. Okay, so we'll push onwards. That Indonesian language version is available, and the medical students could have access to that later on. So we'll just push forward. Okay, so then with regard to the cases within the Indo-Pacific area, every year we see multiple incidents which reach the international press, but this unfortunately represents a vast underrepresentation of what is actually going on. So in this case, a Philippine girl was home from Italy, and she was visiting with her mother, who was Filipina, and they went to a coastal community just for sightseeing and to enjoy their vacation. The child was stung by a box jellyfish that isn't what's pictured here. And in this case, the child was in a great pain, and the boat operator applied gasoline to the girl because he felt that that was the appropriate first aid. So that really caught our attention, and we couldn't believe that that would be recommended. But in cases such as this, where fisher folk are offshore and the adult man gets stung, you can imagine the amount of pain that he must be experiencing to attempt to relieve it with gasoline. Having done so and survived using gasoline, a person can often then presume that their survival was due to gasoline. So this is a basic misunderstanding between the actual practice of science, which is rigorous with control as well, versus anecdotal wives' tales, etc. So because we have such anecdotes and so few medical practitioners aware and available, these things can lead to a life-threatening event and then death. Just be sure, okay. Another event, in fact, the very first one I saw, which was about 10 years ago, was a story from Malal in Mindanao, Philippines. I had the opportunity to go there in person 2 years ago. And this child, again, is very typical of a lot of these stories. A young boy, he's 8 years old, went to a coastal community to swim on an afternoon after school. Unfortunately, he was stung at 2:15 and pronounced dead at 5:45 p.m. So the child was treated with vinegar, but he perished. The news report here claims that vinegar is an antivenom and is something that could treat the sting. I want to show you today data which really explore what vinegar does and doesn't do, but it isn't an antivenom. Classical antivenoms are antibodies that are produced to particular, usually snake antigens, and these are injected. So vinegar obviously is not an antibody. Also, vinegar does not go into the body and reverse or inhibit the venom that's already injected. So there's a lot of misunderstanding in the lay literature, and this really showed me the incredible importance of communicating what I'm doing in the laboratory more broadly. So the dream that I have really, and what's fueled my interest in working in the Indo-Pacific to the greatest extent possible, is to really address this incredible health burden that is occurring in the Indo-Pacific. So essentially, we have morbidity and mortality that isn't being properly addressed. So with regard to an overview of the conceptualization of the team building that I think will be most helpful in this area, I'm showing here public health, field ecology, pathophysiology, and evidence-based care. You might look at these as clinicians and think, how do these interact with one another? How are they related? Actually, this is something that I didn't understand myself as a biochemist for my career. I spent most of my time in the laboratory looking at basic phenomenology. When it came, though, to having made basic discoveries in the pathophysiology of the venom and how they worked, I was then quite excited to try to translate those findings from the laboratory to real public health outcomes. So the ways in which I see these connected is when we look at these tragic loss of life amongst children and we see this public health burden, we can ask who's most at risk, where, and when. We can let various things guide us. At present, it's mostly the international press. But more properly, localities and healthcare providers in specific countries that have this morbidity and mortality health burden can conduct surveys amongst coastal communities. And based upon these surveys, we can really identify high-risk zones and high-risk times of years. So what we found in the Philippines, for instance, is that coastal communities, specifically before the big monsoon rains come in July and September, July to August, before October, basically, there the risk is highest. In areas in which we find, for instance, mangrove estuaries. So that goes to field ecology. So we've let the public health drive where we've gone to look, and in those areas we've looked for what kind of animals are present at that particular time in those localities. And interestingly enough, most all of the localities had this kind of mangrove estuarian coastal zone. And as I say, again, they were at the end of summer before big rains came. So our surveys basically informed us as to where to go to do field ecology. And then having conducted the field work, we can collect the animals that are present and conduct bioassays, such as the blood auger assay, which I'll describe in more detail in the talk. But that can inform us as far as where the serious pathophysiology may be a danger and how these are working, how the various venoms work and various animals work. Based upon that, then we can come up with evidence-based approaches and then ultimately communicate this data back to the local stakeholders. So as you can see, this is an iterative cycle which requires international teams. So I myself am a biochemist, so this pathophysiology piece is really my expertise. But with proper collaborations, I can work with folks in public health, as well as field ecologists, and then we can reduce this to practice with animal models, such as physiologists I've worked with in United States Army and other opportunities that we have to do clinical studies. So the point of it is that we cannot reduce morbidity and mortality or address this major problem without good teams. So I'm really delighted to have the interest of Dr. Vienta and Dr. Munawir and Dr. Tree. The Indonesian component is extremely important, as well as our Philippine collaborators and also in Thailand. So I think we are making good progress, and that this collaboration that we formed and we're starting with now is going to bear fruit, and we will be able to make an impact and reduce the health burden that these animals cause. When we query coastal first aid and clinical care folks, we found that there is a great deal of frustration amongst folks as to how to treat box jellies. They're either very uncertain or have no idea. We do find a certain cohort, though, that answered the survey to say that they were very certain or somewhat certain. I was happy to hear that some folks felt very certain about proper first aid. But then when we queried to ask what exactly the very certain approaches should be, many of these were men, and quite a number of them said gasoline. So the very certain folks could be very certainly wrong. And that presents another cultural issue, because one has to be very sensitive about giving information to coastal folks who have specific points of view that are very deeply ingrained. So that taught me another lesson, that we need to be able to develop really helpful evidence-based communication pamphlets, etc., so that we can inform without insulting or offending folks who may have deeply held but potentially dangerous first aid treatments in mind. Again, the most vulnerable amongst these sting victims are children. And of course, we have, I've been now 12 times to the Philippines. We have the most number of deaths actually reaching the international press from coastal zones in the Philippines. One doesn't have a lot of information from Indonesia, but given the incredible size of Indonesia and that it is an island archipelago, I presume that the health burden in Indonesia could be very high as well. Tagalog Health Station here, a tiny barangay health unit in Samar in the Visayas of the Philippines, had recently suffered a child's death. And we went there to investigate what animals were present. We found the animals and we did the bioassays, and I'll describe that in more detail. I want to introduce my colleague, Dr. Noel Sagil, who was the first person to really help us to find the proper localities, and has done in all 12 of our visits there the lion's share of the work in really connecting us with coastal health workers. But everyone we've contacted has been intensely incentivized to try to address this problem and to come up with systematic ways of testing first aids. Often what has been recommended in these coastal areas are calamansi juice or morning glory leaves. So we in fact tested those in our bioassays, as well as other compounds that I'll discuss later. Okay, so in the Philippines, again, just we're working on network building, public health engagement, field ecology, and education and training. So we would be happy to do more of this in Indonesia, which is basically to go out into the field as we did in Situbondo, to take blood auger plates and to do live bioassays with tentacles. Here are some of the morning glory leaves that we use to test for its efficacy in reducing hemolysis. Today, then, in general, I'm going to present an overview of what are box jellyfish, and then go deeper into the pathophysiology, as well as then to how the pathophysiology informs our first aid and clinical care. And then, as I've already alluded to, what our current research priorities are and how international collaborations can reduce annual deaths in Indonesia, the Philippines, and global hotspots. Box jellyfish, as you may be aware, are cnidarians. They belong to the phylum Cnidaria that includes coral, anemones, and jellies. The swimming jellies, the so-called medusozoans, you may be quite familiar with, but some folks don't realize that the coral and anemone are also in phylum Cnidaria. So the entire phylum is defined by the presence of a specialized cell called the cnidocyte. And cnidocytes contain a massive intracellular organelle called Nidae. And the Nidae take up over 95% of the cell volume of the cnidocyte. There are three different types of Nidae. One is an explosive penetrant Nidae called a nematocyst, and that's the one which literally injects the venom into the body. These are microscopic, and so a tentacle lying along the body can inject hundreds of thousands of these microscopic Nidae into the body. There are other types of Nidae, one that causes the tentacles to be very sticky, and another one that juveniles use to hunt. This is a video that I helped Nat Geo make. I'll play it. The discharge from the surface of the tentacle of hundreds of thousands of these tiny Nidae, which are basically 25 micron in diameter. As it discharges like a hollow tubule that injects the venom into the skin. So obviously that's a traumatic event causing wounding to the skin and to the dermal space and subdermal space. In addition to the injection of the venom, there is an actual wounding, and these tiny tubules remain in the skin. Two main families of the class Cubozoa are the Cryptidae and the Chirodropidae. Both of these families have a four-cornered body plan, but Cryptidae have only one tentacle per corner, whereas the Chirodropids have up to 15 tentacles per corner. So most of the deaths and serious stings that we've seen in Indonesia and in the Philippines and elsewhere are from the Chirodropids. Box jellyfish can see. They have 24 eyes. They don't have a brain, so it's really quite curious. But they have 4 eye stalks and 6 visual apparatus organs on each of the eye stalks. 2 on each eye stalk that have a lens and are capable of color vision and focus, as well as with an iris. And then a slit eye and a pit eye that are capable of near single photon detection at depth. So these animals are quite capable hunters and use their vision to hunt, again, even without a brain, which presents a little bit of a curiosity to us. So now I'll switch gears into the field ecology. Obviously, in order to do my work as a biochemist, I need to be able to collect enough animals to have enough venom to do the experiments that I want to do in the laboratory. This sounds rather straightforward, but it becomes very complicated and difficult when you're dealing with the marine environment. So for instance, when I began my studies here in Hawaii of the Hawaiian box jellyfish, I found that the only time I could collect really healthy live animals was at night and offshore. So this presented a complication for me, but basically we were able to get all the proper certifications to do scuba offshore, and that's how I've done it. A lot around the world, we have cubozoan hotspots, typically near equatorial zones. But again, the biggest distribution is in the Indo-Pacific. So you see here the red, and red is black. Red is from Iroconji sting cases, and black represents death, mainly from the Chiropod species. Right smack in the middle of the hot zone then is Indonesia. So you folks have a massive area of coastal zone with many habitats that are absolutely the perfect habitat for these chiropodids. So for that reason, Indonesia is absolutely center with regard to potential health burden. That said, we don't have a lot of data from Indonesia. We have more data from the Philippines, but again, obviously the Philippines is also an island nation. So I have been now to the Philippines, worked in Thailand, briefly at Java in Indonesia, and then also I've done field work in Australia. Back at home, though, in Hawaii, we have a species that comes to the shore like clockwork 8 to 10 days after the full moon. There are almost 50 species of box jellyfish, and only one species has this unique behavior of coming to the shore 8 to 10 days after the full moon. They come basically to spawn, and then they perish. So each lunar cycle, a particular cohort comes, and we can collect them in these areas here. They actually purposely swim through the cuts in these seawalled areas, and it makes it very easy for us to collect them. But I also work offshore. So this is just as a scuba diver at 1 to 4 a.m., about a mile offshore. And what the jellies do is they come up from their rest of the month, they're living at a depth, and they come up from that area. It's basically a constitutive cyclonic current offshore where they feed. But at this particular trigger point, 8 to 10 days after the full moon, they come up to the surface and they purposely swim to the shore. You can see this animal has this white look. That's because it's replete with gametes, either eggs or sperm. I've also done field work off of Saipan in the Philippines. So I've taken a good amount of time to go wherever we found serious stings and deaths, and to try to determine what the culprit, if you will, is, and where and when we can collect those animals. And so that actually informs our clinical care for local physicians based upon what species are there. So that particular species you saw had only 4 tentacles, so that's a charybdid. Within Thailand, there have been several deaths due to box jellies, and I was by the U.S. State Department as a Fulbright specialist to go and conduct field ecology there. What we did was to go offshore, and I found that these particular animals, unlike the animals that I've seen in Indonesia and the Philippines, they hunt at night, these large box jellyfish. So you can see the amount of safety gear that I use. In this case, I collected these animals as a pre-dive. But these are how the animals look at night. They're actually about a foot by a foot. When their tentacles extend to 10 feet, I grab it and shake it to get them to contract to 1 foot. So this is done with layers of wetsuit as protection But these animals have caused a number of deaths in Thailand. Within the Philippines, the area that I showed you, Tagalog, where more than one child has died, these animals are coming out during the day and the deaths are occurring at the day. I have a GoPro here, basically looking down on the animal. You can barely see it under, and with the sky as the backdrop, you can start to make it out. So this isn't just water that was 2.5 feet deep. Remarkably, this particular animal through my 1 millimeter wetsuit, and I thought that I was protected by a 1 millimeter wetsuit, but it was insufficient to prevent this sting. I had students along this shore ready with first aid, so we enacted our first aid protocol immediately. But you can easily see how these animals then could inflict a lethal wound on the child. Now, in Indonesia, with Dr. Lenta and Dr. Munawir, we were fortunate then to go off near Situbondo and to go off to have a look at the animals and really
Morning, everybody.
hunt in the coastal zone.
Dr. Maharani, Patrick. One seem not to answering. Probably he's still offline.
Hello? Did someone have a question?
No, please continues.
Continue, Miss.
All right, so then this is to show you what we found in North Java. Again, I did this without scuba. In this case, I was just simply doing breath hold surface swims, not even with a snorkel. But going in the shallow mangrove area, which was actually less than 2 feet deep. And this seemed to be the dream habitat for the lethal box jellyfish there. So we can barely see the animals. So here it's almost like a ghost. In fact, I think one of the local fellows told us that that's the name that they had for it. Oops, sorry. Our video failed there, sorry. Try again. Perhaps while that is reloading, we could take a few questions from the students.
Okay, thank you, Prof. And I saw there are some questions from chat. Here, maybe I will work.
So,
Okay.
go ahead.
Could I read the question from chat?
Yes, please.
Okay. Okay, first question from Dr. Faridianto Yudo. In tentacle is assay, why would it use McConkey agar? What the purpose of the assay?
The McConkey agar has intact red blood cells, And the venom contains a component which specifically causes red blood cells to have holes created in them and to lose hemoglobin. So this is a bioactivity assay that we can use with live tentacles.
Oke. Bagaimana Dr. Farid Dianto Yudo?
Yeah, I'm sorry, Prof. Yana Gihara. As we know that McConkey is contain lactose in very rich lactose. Maybe you are adding blood in the McConkey to assay the tentacles.
Yes, the McConkey blood auger, sorry.
Oh, McConkey blood. Okay, okay. I'm sorry.
Okay, no, my mistake there. Should have made that clear.
As I know that McConkey used to identify the Enterobacteriaceae bacteria. Is there any correlation between the tentacles and bacteria?
Actually, yes. So bacteria produce lysins, so streptolysin O and anthrolysine O. And interestingly, all of the Nidaria release a pore-forming protein. So we've published quite a number of papers about that. And it's interesting that there is also hemolytic.
Yeah, amazing. As I just know that streptolysin is produced by Streptococcus, and I just know today. Thank you very much, Prof.
You're welcome.
Thank you.
Okay, thank you. And the second question, dear Miss Angel, this is from Iketut Tirtanandaka, Dr. Ketut Tirtanandaka. Dear Miss Angel, thanks for your presentation. My question is, how the best strategy controlling the jellyfish population, especially at coastal sea, where society get activity? Thanks.
Controlling populations of jellyfish is rather a complicated food web dynamic situation. So in a balanced food web, the various fishes will eat juvenile jellies and feed on the planulae that are developing along the mangrove roots. But when there's overfishing and depletion of these, both the larval and juvenile stage fish that would normally eat these juvenile medusae, one can see that more jellyfish make it to adulthood. So it's a little bit of a negative cycle in that when the food web is disrupted by overfishing, jelly numbers can basically increase. So if one wanted to reverse that and control jelly numbers and reduce those populations, then perhaps more protected coastal fisheries, as well as other animals that eat jellies, turtles, etc. But in most coastal areas in which there is a good amount of economic pressure for fishing, the fishes have been depleted. So this is basically restoration of the marine food web would reduce the numbers. Other than that, folks have tried to use protective nets to prevent jellies from being in certain areas, but this doesn't always work because oftentimes the juvenile jellies can go through the mesh of the net and grow up and then cause havoc. So it is a complicated question.
Thank you. Silakan Dr. Tirka.
It's all. Thank you very much, Professor Angels.
And then the third question. Hello, Prof. Yana Gihara.
Yes.
Hello, Prof. Yana Gihara. What is the best way to educate coastal population? And are there any specific suits for divers that offer protection against things? Thanks in advance.
From the standpoint of protection for the diver, I found that multiple layers. So basically, I wear 3 layers. I have a lycra layer, and then a 2 to 3 mil layer, and then another layer on top. this is perhaps overkill, but given the physicality of having to collect the animals and swimming movements, etc., I want to be very safe. So that's been my approach. And sorry, what was your other question?
The best way to educate coastal population.
So that is clearly out of my lane, as you might say. I'm definitely the bench biochemist, the lab rat scientist. But I have worked now more and more with folks whose expertise is public health. For instance, Dr. Catherine Perkle came with me to the Philippines. So I presume that expertise amongst your public health folks in Indonesia would really help us to create informational products that are accessible to coastal fishermen. I think Dr. Rianta did a wonderful job in outreach when we went to various communities along the islands there, and she sat with the fisher folk and spoke with them one-on-one. So I think the communication is extremely important and is the most critical part of this whole iterative cycle, how to communicate these findings back to the fisher folk.
Okay, thank you, Prof. And there are another question, but would you like to continue the presentation or answer the question? Or maybe
Well,
take a break?
Is there one more question and we could take a break then?
Okay, one more question from Dr. Winta Dias-Fitri. Aloha, Angel. May I know whether box jellyfish in Hawaii and other parts of the world have the same or similar venom characteristic?
Yeah, so that's a fascinating question, and that's really driven a lot of my work. And there are folks who have presumed that every single jellyfish will have a very different and unique mixture of components that comprise its venom. And while that is so in a particular sense, in a more general sense, these venoms are highly conserved. So every venom that I've looked at, for instance, will have a pore-forming protein, will have various lipases and proteases and other components. So within the phylum Nidae, it seems that the general paradigm of what constitutes a venom is highly conserved within the whole phylum. That said, from species to species, the particular proteins that that animal uses differ as far as the basic amino acid structure and the bioactivity. So the most potent of the pore-forming proteins, the fastest acting ones, are in fact from the chirodropids. So the venom of the Hawaiian box jellyfish has a pore-forming protein, which I'll describe in our next section of the lecture, but it is the Hawaiian box jellyfish pore-forming protein is nowhere near as potent as the chirodropid porin. So we've done these experiments in piglets, for instance, and one sees that the dose required for death in the piglet from the chirodropids that one finds in Indonesia and the Philippines is basically 1/100th of the dose required from the animals we have in Hawaii. So they're far more lethal, but they are the same family of proteins that drive the pathophysiology.
Baik, Dr.
Thank
Winta
you.
dan Kelvin, bagaimana? Apakah ada yang ingin didiskusikan lagi? Ataukah jawabannya sudah cukup? Begitu, Dr. Winta,
Sudah
Kelvin?
cukup. Thank you very much.
Okay, thank you, Prof.
So maybe we'll have a 5-minute break and I'm going to reload my computer because it's doing that little swirling problem.
Okay, Prof.
Okay, I'll be back.
5-minute break. Okay, thank you.
Batiknya Prof Yana Gihara bagus ya.
From Indonesia.
Iya, Bu Dekan.
Itu mungkin Dokter Winta dulu beli di mana itu?
Di Madura.
Di Madura. Oh, Oh, bagus loh. Cantik itu.
Beliau milih sendiri. Beliau suka batik.
Dia suka batik.
Iya.
Oke.
Pulang
Oh,
dari Sampang itu budekan, mampir.
Mampir ke.
Iya, batik
Bagus
tulis
ya?
di tepi. Iya.
Ya bagus itu. Dijahit di sana berarti ya, di Hawaii.
Beliau menjahit sendiri.
Oh,
Beliau
jahit
menjahit
sendiri.
sendiri, iya.
Oh, hebat ya. Mungkin kalau di Hawaii tailor itu nggak kayak di Indonesia ya. Penjahit itu sulit kali ya. Hebat sekali.
Kalau di jalan beliau bawa itu knitting budekan. Yang apa itu? Knitting itu, ya beliau bawa itu. Kemudian beliau bikin sweater, bikin ya.
Wah, tangannya juga hebat sekali ya. Ini
Halo,
tadi
Bu
ada
Dekam.
pertanyaan. Ya, halo Pak Munawir. Halo, apa kabar? Wah,
Baik,
sehat ya, Pak,
alhamdulillah.
ya? Di mana ini,
Iya.
Pak Mon? Di mana? Di
Iya, ini
Surabaya apa di-
vaksinasi nih. Vaksinasi.
Vaksinasi. Oh, jadi vaksinator,
Iya.
Pak.
Screening.
Oh, screening. Waduh, hebat. Hati-hati ya, Pak ya. Tetap jaga kesehatan.
Nggih.
Nah, ini kenapa
Nggih,
maskernya
nggih.
cuma sederhana begini?
Oh iya.
Enggak double masker atau pakai N95. Susah ya cari N95 sekarang.
Enggak, ya. Ada 95 ada.
Iya,
Iya,
harus
Prof.
jaga
Angel
kesehatan.
memang luar
Luar biasa
biasa.
ya.
Saya kan ke sana, ya. Di rumahnya itu lengkap itu ya. Jadi kayak apa itu menjahit itu lengkap itu.
Oh, di
Tempat
Hawaii
motong,
sana.
apa mesin semuanya.
Di Hawaii sana.
Ya, di rumahnya.
Oh, Pak Munawir sampai ke sana ya dulu ya.
Iya.
Ini Prof
Iya,
Supartono,
tahun 2019.
Oh ya. Prof WD1 Dr. Winta. Prof Supartono ngendikan kalau kita bisa melebarkan kerjasamanya tidak hanya FK, tidak hanya Fakultas Kedokteran, tapi barangkali juga
Saya kira Fakultas Biologi
kerjasamanya,
juga saya kira.
ya mungkin juga oceanografi atau apa yang terkait
FTIK budekan bisa.
FTIK, ya nanti mungkin kata Prof kita bisa melakukan kerjasama yang lebih besar dan mungkin ya kalau perlu kita tinjau lah bagaimana sih di sana bisa kita bekerja samanya gitu.
Iya, dan mungkin dengan beberapa universitas di Filipina yang kerjasama dengan Prof. Yana Gihara juga budekan. Ini ada beberapa mahasiswa beliau yang hadir di sini juga dari Filipina. Iya, dari Filipina. Iya.
Mana nih? Ada yang berkenan berbicara? Maybe from Filipina? How are you? From Filipina ya. Dari Jember ada nggak nih dari Jember juga?
Dari Jember enggak ada, hanya Dr. Munawir saja. Ini dari Filipina sepertinya Sofia de Guzman.
Mahasiswa lagi libur nih, Bu.
Cirwin Fernandez.
Pakai
Hello.
Pakai Zoom Pak Munawir. Ya, hello.
Iya, pakai Zoom.
Ya, from Filipina. Ya, tadi di Filipina bagus ya, aktif ya.
Iya. Kemudian ada Dr. Tri Maharani dari Indonesian Toxinology Association. Dari beliau juga penasehat WHO untuk gigitan bisa ular, budekan. Dr. Tri, halo. Mungkin kita juga bisa bekerja sama, budekan. Dan beliau juga
lanjutkan.
Halo Bu, saya Dr. Maharani.
Iya dokter.
Cuma saya lagi ada 2 meeting nih ya, yang satu dengan Rek Asia dan yang satu dengan teman-teman nih. Saya sebenarnya sering diminta tolong oleh teman-teman Hang Tuah untuk ngomong tentang gigitan hewan berbisa, terutama ular. Biasanya Dokter Angel yang meminta tolong saya. Angel spesialis emergency.
Iya.
Saya kebetulan spesialis emergency juga. Iya. Untuk kasus-kasus jellyfish, memang sebetulnya karena saya clinical toxinology ya, saya memang sudah mengumpulkan kasus itu mulai tahun 2013. Hanya saja memang fokus saya karena kondisi Indonesia untuk kasus gigitan ular itu angka fatalitasnya itu sangat tinggi. Jadi memang di tahun-tahun ya 2010 sampai sekarang ini saya fokus ke gigitan ular, meskipun jellyfish ya tetap ada. Nanti saya kemarin saya sudah sharekan ke Dr. Winta ya untuk jurnal terbaru saya di bulan Juni kemarin ya, tentang blue bottle jellyfish yang saya kumpulkan dari tahun 2019 dan 2020. Jadi ya monggo silahkan, karena memang saya clinician, jadi kerjanya di rumah sakit.
Dokter.
Ya. Terus sekarang nih saya jadi periset di pusat 2 Litbangkes Kementerian Kesehatan. Nah sekarang ini kan BKPK nih namanya ya. Nah jadi kalau untuk kajian kemarin saya sudah mengajukan kajian untuk tahun 2022 tentang jellyfish yang blue bottle. Jadi kalau misalnya teman-teman dari Hang Tuah mau kerjasama dengan kami, monggo teman-teman Litbangkes senang sekali ya. Barangkali ada teman-teman S1,
dokter.
Ya, teman-teman S1 yang mau membuat skripsi atau tesis, monggo untuk. Tapi memang saya lebih ke arah sebagai clinician ya, jadi mungkin teman
nanti
dokter
sama-sama,
kalau
dokter.
saya
Sama-sama nanti dokter. Sama-sama karena visi misi kita juga kelautan, pastinya kita lebih ke arah jellyfish ya. Syukur-syukur box jellyfish ini.
Ya.
Mungkin nanti kita progres antara UHT dengan yang lainnya.
Iya, kalau saya sih karena sekarang sudah menjadi peneliti di Litbangkes dan beberapa kajian saya sudah masuk, monggo kerjasama dengan kami di Litbangkes Kementerian Kesehatan. Tapi ya memang klinik sifatnya medis ya, jadi bukan yang identifikasi jellyfish-nya bukan, tapi ke arah medis, misalnya pengobatan, sign dan simptom dan sebagainya.
Nanti pasti arahnya akan ke sana, dokter. Ya kan? Kita menemukan sesuatu, arahnya untuk kehidupan apa yang membantu untuk manusia supaya
treatment
selamat
dan ngatur. Ya semoga
nanti ke depan.
Bisa membantu Indonesia ya, karena kasus-kasus jellyfish itu kan banyak yang mematikan juga seperti kasus gigitan hewan berbisa. Matur nuwun dokter untuk
terima kasih Dokter Maharani. Terima kasih, monggo silakan dokter. Maybe Prof. Yana Gihara will continue.
Okay, thank you. So the next section then will really start to address some of the questions that you folks have already raised. So let's get into the real heart of the matter, the pathophysiology and the venom biochemistry. So when a person is stung, basically, as I showed in that graphic animation, hundreds of thousands of these microscopic Nidae explode. The penetrant nematocyst is shown here compared to the size of a human hair. The tubule is hollow, so it acts like a hypodermic needle to inject the contents of the nematocyst capsule. And these Nidae tubules have different lengths. The chirodropids have the longest tubules of any of the species of the within the phylum Nidaria. So these nematocysts from the chirodropids basically can inject up to 2 and sometimes 3 or 4 millimeters deep. So it's a very deep wound that can occur. They also are very large compared to other species. Now, the critical question that started me on my process here 24 years ago after I was stung, I looked into the literature to figure out what caused such a horrific sting that I suffered and the respiratory insufficiency and the whole sequelae, which actually constitute Iroconji Syndrome. So the first thing I found was that within the literature, there was a great mention of these lethal stingers from Australia, and they were far more horrific than the animal that I encountered in Hawaii. So I was grateful that I hadn't been stung by one of these. But the prevalent hypothesis at the time was that these venoms worked by having a silver bullet type of ion channel blocker, which makes sense given the clinical presentation of acute cardiovascular collapse. One might presume that there is some kind of a paralytic peptide or channel blocker that underlies that event. So in the next few slides, I'm going to share with you a little bit more about the physical trauma that nematocysts, the penetrant Nidae, can inflict, show a little bit more about the explosive discharge, but then really get into the biochemical characterization of the venom. Basically, as a biochemist, this was my path, which is a pretty straightforward and a routine approach that one would take towards any unknown, to basically try to characterize what components comprise a complex bioactive substance, and then to elucidate the mechanism of action of each of the components. Then ultimately to try to identify, is one or more of the components lethal? Can they in and of themselves recapitulate death in a test organism? And ultimately then hopefully to develop particular inhibitors for any of these lethal components, and finally to develop animal model of any potential useful inhibitors to test whether or not it could be an effective therapeutic. I'll go ahead and just give you the short version of this 24-year story, is that yes, we found specific inhibitors of the fastest acting lethal agent, and we have been able to reduce this to practice into a now fully approved over-the-counter type of cream that rapidly inhibits the venom and prevents the sequelae. So this is quite a happy story for me. Having said that, though, it's all a dose-dependent outcome. And at high enough doses, the inhibitor that we have may or may not be able to stop that locomotive, if you will. So it's very imperative that we continue this work and look for other ways to augment this kind of inhibitor with protection of the airway and breathing and heart function, giving the inhibitor time to work. So there is critical care that needs to occur in addition to any particular inhibitor that I have developed or that we could develop in the future. So all that is to say that this is a clinical team kind of effort, but for now we're going to kind of go into the biochemistry. So the first thing I did when I started working with these animals was to develop a way of isolating the venom. And then I looked at whole blood. It's actually a drop of my own blood, and a time-lapse microscopy to see what goes on. So within the very first frame already, you could see crenellation of the human red blood cells. The platelets have already aggregated, and white blood cells start to develop massive pathologies, including foam cell formation. At the end of this 8-minute time series of microscopy of the live blood, one sees all these red blood cell ghosts. So basically, we started with intact red blood cells, which quickly had these spicules of crenellation. The platelets were actually the first domino to fall with that aggregation. So within whole blood, the venom is causing a good deal of pathology already. And this was not consistent in my mind with the hypothesis that was prevalent in the field that the venom worked through an ion channel blocker. What one sees in this time series is more consistent with some kind of a massive cytolytic component. So I didn't put aside the idea that ion channel blockers could be playing a role, but I was alerted immediately to the presence of some massively cytolytic component within the venom that would be able to cause the loss of hemoglobin to the production of red blood cell ghosts, as well as these pathologies in the platelets and white blood cells. In order to do the work, though, the first thing I did was to follow methods that were published, and the bioactivity of the venom that was produced using those older methods was far less than would make sense that the venoms could cause death. So basically at the time I started this work, folks were collecting tentacles, freeze drying them, then macerating them in saline and taking that saline extract as venom. And you could quickly imagine that once you freeze dry a natural cocktail, you may or may not bring everything back into solution. Certainly if there are lipid type compounds that comprise the bioactive components, you're not going to recover those with a saline extract. So I went about to develop a method to isolate intact Nidae, the capsules, from live tentacles. And we've been able to do that now in the field with Dr. Noel Sagiol in Lucena and in Samar, and with Dr. Wienta and Dr. Munawir in Indonesia, and then with others in Thailand. So we're able then to go out, collect a live animal, clip the tentacles, and put them in citrate. So 1 molar trisodium citrate. Basically the mesoglia of the tissue of the tentacle then contracts just a tiny bit, and the Nidae slough off completely intact, undischarged, and we can spin that, centrifuge that, and collect the Nidae free of the tentacles. In my laboratory in Hawaii, I then rupture them with a high pressure cell and then snap freeze them in liquid nitrogen under gaseous nitrogen. So we can see then that the tentacles are comprised of these Nidae, in this case, a particular type of uretial in the Hawaiian box jellyfish. These are undischarged. Here's how a discharged Nidae look. So the undischarged Nidae contain this physical apparatus, which is the tubule, as well as the venom. When we spin and pellet the Nidae, then we can rupture them en masse at very high pressures and then quickly snap freeze that. So we have the complete content. It isn't an aqueous extract. It is complete content of the Nidae, which turns out that to be about 30% lipid. So it's a very lipidic cocktail, and a rather slow release kind of event upon a sting. So that was surprising. Again, you saw in the time course I showed you just a moment ago, evidence of various pathologies in platelets, white blood cells, and the red blood cells. But when we did electron microscopy of the red blood cells themselves, what we see is at high magnification, these tiny ring-like structures, which reminded me of streptolysin O from bacteria. At higher resolution, we can see that these are very stable rivet-like structures. Basically, what we found then is that the venom contains a monomer, a 42 kilodalton monomer, this pore-forming protein, which self-assembles, and here's a partially assembled porin, so it's a multimer, so like a string of pearls. And it has a calcium binding domain that has to find one another and form this pore, this multimeric pore, in order to have a functionality. Once that multimeric pore is formed, however, though, we have basically a hole in the red blood cell. And these porin rivet-like structures are too small in the inner diameter for the release of tetrameric hemoglobin in the beginning, but they are not too small for potassium to leak out. And the red blood cells carry a lot of potassium in contrast to the plasma potassium, which has to be very low in order to maintain electromotive force throughout the body amongst cells. So if one has holes being formed in red blood cells and such that potassium can leak out, you can immediately see that you might be in danger of hyperkalemia. So when I first observed this electron microscopy, it struck me that a potential alternative hypothesis to the ion channel blocker that had been put forward in the literature could be that there is cardiovascular collapse due to hyperkalemia, which at high enough levels of potassium in the plasma, basically the cardiomyocytes can no longer beat. So this is just another picture to kind of illustrate that. So again, this is a multimeric pore-forming protein and very similar in structure to anthralysin O and streptolysin O. So it's very curious that this is conserved throughout the animal kingdom from bacteria to Nidaria, and this is actually coded for in the genome of the Nidaria, so it isn't from a bacterial contaminant per se. And again, it's too small at the beginning for tetrameric hemoglobin. But these things do self-assemble. And then we studied the self-assembly and hemolysis at different temperatures and found that at higher temperatures, even as low as 45 degrees Celsius, which is a safe hot bath, that this self-assembly could no longer occur. And this time period required for self-assembly is delimited. So that led us to consider whether or not this could be a type of first aid to offer transient heat during this critical phase that the venom's been injected to prevent the self-assembly, which can drive the development of these pore-forming proteins, which then cause various sequelae after that integration into the red blood cells and other cells. And so we found, in fact, that this is quite reproducible, and it isn't anywhere near what's required for complete denaturation. Obviously, it isn't 100 degrees Celsius and SDS, etc. So we have basically just inhibited this potential calcium binding domain from finding one another. So this was actually a very useful discovery. And this is all published, so we're happy to share with you that published data. So this is kind of another approach we found towards inhibiting the self-assembly of the porin was through other divalent cations. So having found that calcium was required for the self-assembly, we went to query various other potential inhibitors. So I went through a number of divalent cations, including zinc and copper. And in this red blood cell hemolytic assay, if you will, red is dead. So the way the experiment is done is we take red blood cells in solution and add a various dilution of the venom. And at undiluted venom, basically all the red blood cells rupture. So when we spin down and take the supernatant, so these are all supernatants, the supernatant is red because it has the hemoglobin. And this is in 0 millimolar zinc gluconate. As we go up in the potential inhibitor, we find, in fact, inhibition. So in this case, this is a very powerful inhibitor of the self-assembly of the pore-forming protein. So we investigate that as a potential therapeutic modality to inhibit venom. Unfortunately, we found that the zinc, while it inhibited the self-assembly of the pore-forming protein, it actually increased the activity of a zinc-dependent protease. So it was a double-edged sword, and we abandoned zinc. We found that copper actually worked at about 1/50th of the concentration, and we found no negative activation effects of the copper. So we went forward to develop potential first aids based upon the copper gluconate finding. And this is just to show you how interesting the venom is. This is a HPLC, which is size exclusion HPLC. And I've done it in an aqueous environment so that everything could be taken fraction by fraction to do immediate bioassay. And in this case, we have the peak A and the peak B. So the peak A are very large proteins and protein complexes. And what we found is that this is mostly comprised of lipases in a milieu of bioactive lipids. So the venom, as I mentioned to you, was highly lipidic. all of the pore-forming protein, the porin is in this tiny peak. And these are other small molecules, peptides, some other interesting things. But when we inject it into a mouse, just this tiny fraction, which is less than 1% of the total biomass of the venom, we had the exact rapid recapitulation of death. So it appears that the heavy lifting of the pathophysiology is all due to this porin. I still don't discount that there could be bioactive peptides that are ion channel blockers amongst these that we haven't yet discovered. But for all intents and purposes, in all the work thus far, in mice as well as in anesthetized piglets, the pore-forming protein is the fastest acting agent and able to drive all the pathology that we've seen. And towards the question that was asked earlier, when we do this kind of size fractionation of chirodropid venom, other box jellies besides the local Hawaii jelly that this is, the Alatina alata, we also find this kind of peak A, peak B behavior, and we find also the porin. When one does the amino acid sequencing, though, they're different and distinct species to species, but the components in general are highly conserved in terms of the classes present. But then again, as the questioner asked earlier, the potency of the chirodropid porin is far higher than of the Alatina porin. So there does seem to be some hierarchy in toxicity that is driven by the porin itself. But that said, the porins from the chirodropids are also inhibited by copper gluconate over the same dose range. So this is just showing the inhibition of self-assembly in the presence of copper. And here we can actually go down to 20 micromolar copper versus zinc, which requires higher concentration, but had a negative effect of accelerating the activity of a different component. So we rejected zinc, but we find no negative side effect of copper. So copper or the 45 degree both inhibit that self-assembly step. So this is published work. I'm happy to send you PDF if you don't have it in your readily accessible. But basically, you will immediately recognize pulsus electrical activity. So we see time sweeps here with the Doppler sonogram that the left ventricle ceases to have its contractility, whereas the electrical conductivity is still intact. And we have the typical hyperkalemia kind of EKG read here. So we were able to rescue lethally injected mice using the inhibitor. And for the medical students, I would challenge you to go back to your undergrad and look up the Nernst equation, basically to kind of understand what goes on here. So I mentioned to you earlier that hyperkalemia could stop the heart. How does that work? Well, basically, we know from the Nernst equation that in order for the ATP generator to continue working so that the heart can beat, one needs to have a higher intracellular potassium and low extracellular potassium. There are other ions involved, but for simplicity's sake, I'm just showing you the potassium. So if massive amounts of red blood cells then have these porins and lose their intracellular potassium, and this leaks out into the plasma, we can have elevated plasma. Basically, clinically, you're going to be looking for anything over 5 millimolar, but just for dramatic sake, I wrote it here to the 15 millimolar, which clearly is a lethal amount of plasma potassium. In this context, the resting membrane potential drops from the required 96 of the cardiomyocyte to 61. And at 61 millivolt, you don't have enough resting membrane potential for the heart to beat. But you'll notice, if we will, this is a dam, and if we make that analogy, but there's no holes in the dam. So the heart muscle cell its membrane hasn't been perforated or affected. It's just a transient external level. So this is reversible, the good news is. And we provided this information in the Philippines, which led to some life-saving approaches by barangay health workers, so that one understands that if you can buy enough time to bring the potassium back down again through heroic CPR with rescue breaths, for instance, so that the kidneys can clear the potassium, then the heart could restart. So you may have this kind of apparent cardiac death, which is simply due to this external high potassium, so pulseless electrical activity. That is in contrast to authentic cytolysis of the cardiomyocytes, in which they actually are perforated and leaking their internal contents. And of course, in that case, it would be an irreversible situation. The fact that that it is reversible, and then what we've shown earlier was good news, and helps inform clinical care. So to put all that together in kind of an overview schematic, if you will, the venom porin can cause holes in red blood cells, causing the loss of intra-red blood cell, intraerythrocyte potassium into the plasma. The plasma then can cause this kind of, the plasma potassium then can cause this transient loss of electromotive force because of the change in the resting membrane potential to cause pulseless electrical activity and death. So this, so far, is the hypothesis that has been supported by the data. Again, I'm open-minded. If we do find a ion channel blocker, we'd be happy to work on that too. But so far, the data are actually rather different than the PET hypothesis that I read about 20 years ago. And this is another schematic just to kind of put everything together for you. Now, with the basic timeline here, so you have an idea of how quickly these things happen. So within seconds of a sting, enough venom can be injected, and these porins can assemble within seconds to form these kinds of rivet type structures so that potassium can leak out. So there can be, within 5 minutes, catastrophic cardiovascular collapse. And we've seen this is a dose-dependent outcome in piglets. So at high enough doses, which is actually a very small amount, we have an acute rapid death. At lower doses that aren't sufficient to stop the heart, one can still have serious sequelae, including what's called Iroconji Syndrome, which I'll describe a little bit later. But basically, the symptoms of that include headache, anxiety, sweating, a tachycardia phase with hypertension, and then followed by bradycardia and hypotension. So there is also, of course, the dermal response. So all of these things occur in series, and at a high enough dose, it's just game over. But at lower doses, there's still a window in which deaths have occurred between 4 and 48 hours after a sting. Another representation of what goes on here is in this schematic, showing the capsule and the tubule with the spines. Basically, the venom, the tubule starts to unwind, and so it actually injects the venom deep and potentially into capillaries, but also it unwinds, and the venom, because it's highly lipidic and kind of a slow release mixture, is deposited within the entire dermis and can affect dermal mast cells and other immune surveillance cells to cause release of cytokines and to initiate diapedesis and massive site inflammation, as well as direct damage to the sensory neurons, which kind of explains that burning pain that most folks describe. And you saw on the time series of the whole blood that the platelets are also affected. The platelets are sequestering catechol, they sulfate and take them back up again to have a short lifetime in the plasma. So when massive amounts of platelets are disrupted, you can drop all of those storage catechols also into the plasma, which starts to explain what's been defined as comprising Iroconji syndrome, which is a cytokine storm and a catecholamine surge. And then again, we've kind of looked at that from the standpoint of in the piglet, and we see that the hypertensive and followed by the hypotensive phase track with the release of these catechols, sequestered catechols from the platelets. So the white blood cells you saw also being affected by the porin, they immediately release the stored, ready to go granules of various cytokines, and those cytokines go into the bloodstream and affect naive white blood cells to start producing cytokines. So suddenly you have what is constitutes a cytokine storm, and it looks very similar to sepsis with the cytokine storm and the catecholamine excess. The histamine overload is another part of the arms of the pathophysiology. So basically, the biochemistry has really illuminated a lot of these different arms of the pathophysiology. A different representation of what I just said, the porin inserting into the membrane, whether it be red blood cell, white blood cell, or platelet, leading to another round of pathology to basket cells, et cetera, degranulating cells. So we've done time series of blood smears with stains in both the chirodropid and the cryptidid venoms, and we see the same kinds of things. So it starts to make sense as far as the clinical picture. Another part of the clinical picture is capillary leak syndrome. And in the interest of time, I won't go into that in detail, but that also fits together with the components. And just to kind of put this out there, and we could discuss this in more detail at another point. So altogether then, if you want to look at all of the things we've looked at in the venom, the lipases, the bioactive lipids, small molecules, we find that these drive another arm of inflammatory pathophysiology. So presently, this is the short list of therapeutics that I'm studying with the animal model here. We have had obviously a great discovery with the copper gluconate as a potential inhibitor of the venom of starting this, but a lot of the folks will appear at clinic where they are actually farther down the track. So the pharmacologic interventions cannot undo, the copper will not undo what damage has already been done in the disease course. So in terms of different time points of intervention and what might be useful or not, these are some of the potential approaches that we're studying right now. So what does all this mean for first aid and clinical care? If you look on a lot of websites, even Mayo Clinic in the US, there's a lot of horrible advice, from scraping stings with credit cards to applying ice. And then there's the popular notions that there are other things that one can do, gasoline or shaving cream, etc. So we developed, as I mentioned in the beginning, various models to test these particular prevent or inhibitors of progression of the sting. Do they help or do they make things worse? So you've seen already the 96 well plate-based assay, that's the hemolytic assay, but then we also have mouse injection and piglet injection, but more of interest to me are tentacle-based assays. So rather than starting with a venom that I've purified in the laboratory, which may or may not have all of the bioactive components in it, I would rather start with a live tentacle from a healthy animal that's just been collected from the field. So the blood auger with the intact red blood cells in the auger plate provides a fantastic tool for looking at tentacle-driven activity. So when we lay the live tentacle on the blood auger, it spontaneously stings, and we can count the number of Nidae that discharge and compare it with what happens in a human skin situation. And then we've also done live tentacle studies with pork, with freshly slaughtered pig skin, as well as with live anesthetized piglets in the time course in a surgical suite. So what do we find? Well, basically we found that vinegar is a good way to prevent things from getting worse. It basically is a weak acid, and it fixes this complex architecture that makes up the capsule and this little trap door that has to open, so-called the operculum of the nematocyst. So when the tentacle touches the skin, only a small number of Nidae discharge at the moment. A large number of these capsules are left sticking onto the body undischarged, like ticking time bombs. The problem is that even though they don't discharge immediately, they still can discharge. So rubbing sand, any site pressure will cause them to discharge, or cold fresh water can cause discharge, etc. Whereas when one floods the site with vinegar, these capsules become altered structurally such that they can never discharge. This is true for the cubozoa. I should mention that there are some scyphozoa in the Mediterranean which don't follow this rule. But for these lethal chirodropids and cryptidids, we have yet to find an animal where the vinegar is not irreversible inhibitor of discharge. Okay, so now this is showing you the blood agarose. I have kind of refined that to use low melting point agarose and taking human blood, red blood cells, and mixing that with low melting point agarose at 37 degrees, and then letting that chill onto a microscope slide. So we have a very thin layer of red blood cells here. There's a tentacle laying on top of that. So this clear zone then is that zone of hemolysis. And this method is published, and we have videos of the methodology if anybody would like to see the videos. We can also amend that by adding a barrier function with a, this is small intestine, pig small intestine that's sterilized and basically then rubbed with anhydrous lanolin so that it has a waterproof barrier function. So we lay the live tentacle on top of that. It can sting through this lanolin covered membrane and then insert the venom into the red blood cells and cause lysis. Then we can spritz the outside with vinegar, things we couldn't do directly onto the agarose itself. So as I mentioned, we had counted the number of Nidae that are fire over human skin versus over the agarose, and these are equivalent. So it's very interesting that the spontaneous discharge, something about the milieu of the red blood cell agar causes the discharge just as it does on human skin. And with higher magnification, you can see the individual Nidae that are discharging the venom. So this is with a black background, so the red blood cells here are intact, and where the tentacle had laid, you see the zone of hemolysis. So using that, then we can query, what does scraping do? What does rinsing with seawater do? What does, all these different potential treatments do? So for instance, ethanol, etc. And we can see that scraping is a mistake. And you might imagine that it makes sense, because if you have an intact, fully functional tentacle and you scrape it off the body, you're basically going to roll it down the skin. It's still able to sting, and the Nidae that are left sticking onto the body can fire off. So each of these little white zones here in this is more wounding and a higher venom load. So pulling off gently was okay, but vinegar was better. Seawater sounds okay, but actually it also is worse, and it's just kind of globally worse because the seawater is a do no harm for the tentacle, and again it's just rolling down the body and more of the Nidae can fire, so there's a higher load of venom and more hemolysis. So we use that kind of a model then to really go through particular potential first aids, some that are available in the US over the counter, such called Safe C, and then the copper gluconate based technology that I was reduced to practice by the University of Hawaii School of Pharmacy here. Compounding pharmacists took our work and then prepared a cream, a Stingnomore called cream, and Stingnomore spray. So the hemolysis after the use of the Stingnomore cream, either a preventative or as a post spray, massively reduced the hemolytic zone in these test blood agar slides. Now, we also took freshly slaughtered pig skin in the Philippines because we wanted to look at gasoline, and obviously we would need a real skin in order to query gasoline, because that small intestine membrane would be no match for gasoline. So we prepared this kind of a field-friendly little, so basically using hardware store window screen edges with the side bits, and clamping on freshly slaughtered pig skin, and then putting on live tentacles caught immediately that day, and tested various treatments. So here's the control, and you can see the wounding. So the chirodropid tentacle created a massive wound that you can immediately see with the naked eye. We also then compared that with vinegar as a treatment to pull it off, to wash it off, versus Stingnomore spray. And this actually gave us the best result, as far better than even the vinegar, to remove the tentacles. And then after that, we used the cream, and that gave us a very good result, versus the gasoline, which was just a horrific worsening of the injury, and also kind of a liquefaction of the body, of the fat within the dermal space. So one can imagine that that would be a horrific injury for a child. Then we've also done these kinds of work with the live anesthetized piglets, again, adding live tentacles to the animal, and we're doing all kinds of physiologic measurements over 8-hour time courses. And then at the conclusion of the experiment, we then sacrifice the animal and take the various organs to do histology and look at the outcomes. Okay, so to conclude, the current recommendations based on the evidence that I've shared with you today is basically to rinse off the tentacles with vinegar, or if one had the Stingnomore spray, and then to soak the area in 45-degree water for 42 to 45-degree water for 20 to 45 minutes, or use this Stingnomore cream. And then avoiding scraping, don't apply ice. This again is just another picture of that. So this technology is granted a full US patent. It inhibits the pore-forming protein found in jellies, but also it potentially has some application in sepsis and other disease states that we're working on. We've queried the efficacy after humanitarian donation in various areas. And we found it to be highly efficacious. So thank you for your attention, and I open the floor for questions.
Thank you, Prof. Yana Gihara. Okay. There are a lot of questions in chat room. I will read it one by one. And one question came from Dr. Tamam. Okay. Hello, Angel. According to your study so far, is there any chance that jellyfish toxin can be used as life-saving drugs or medicine?
That's a good question. And I think it could be that there are really useful bioactive peptides that they themselves are not toxic. So for instance, within cone snails, they produce a broad array of really interesting peptides. I haven't focused so much on looking for those activities, but I think that the possibility is quite high that there could be really interesting peptides that are also components in the venom.
Okay, thank you, Prof. Bagaimana, Dr. Tamam? Or maybe
yeah, thank you very much, Angel.
Okay. And then the next question from Tiffany Taviana. Good morning, Mrs. Angel. We know that the medusa form have the ability to sting us, but this polyp and ephira, young medusa form also can sting us. Thank you.
Is that a question?
Yeah, asking about how about this polyp or ephira, young medusa form?
Oh, yes.
It also can sting us.
Yes, they do have nematocysts. So the ephira, but in this case, the cubozoa strobulate to directly go from the planulae to the juvenile medusae. And that juvenile medusae, as well as the planulae, do have Nidae that are functional and capable of discharging.
Okay, bagaimana Tiffany, Taviana?
Yes, I understand. That is actually very clear. Thank you, Miss.
Welcome.
Thank you. And then another question from Annahel Hakim. This is our student too. Good morning, Prof. Yana Gihara. It is an honor to attend your lecture. In the beginning of the presentation, you show some picture of sting marks. How fast could that mark appear? And does putting pressure, like tying up the area, would help to slow down the venom from spreading and cause systemic effect? Thank you.
That's a really good question. And originally, folks considered these venoms to be like snake bites, and they considered that compression bandages would be a good approach to slowing down the so-called spread of the venom. Unfortunately, the outcomes are completely contrary to the intuition here. And now we can kind of understand perhaps why, because any site pressure will just cause more discharge of the venom. It isn't the same as a snake, which will inject milliliters into a particular space, and you could basically isolate that space from the circulation. In this case, the Nidae are injecting deep into the tissue, into the capillary bed, and any pressure will basically increase the amount of venom that's being released. So it's counterintuitive.
Oke, Rahel, bagaimana? Adakah yang mau ditanyakan lebih lanjut? Atau sudah cukup jelas?
Sudah cukup jelas, dokter. Thank you so much, Prof. Yana Gihara.
You're welcome.
Okay. This is another question from Dr. Budi Utomo Wiono. Good morning, Prof. Yana Gihara. During the dive, can we avoid from the box jellyfish sting by wearing the swimsuit or wetsuit, including gloves and socks? Thank you.
So that was a rude surprise for me in the Philippines, that the chirodropid is perfectly able to penetrate the 1 millimeter neoprene lined wetsuit. So I had to go to 2 to 3 millimeter. So clearly the Lycra socks would be insufficient. It's curious because in Australia, they typically use what's called a stinger suit, which is just a Lycra kind of bodysuit. And in the past, they had actually even used women's support pantyhose. So it remains to be identified, but the species that are prevalent in Indonesian waters and Philippine waters may not be the same as the Australian species. And in my experience, they're much more profoundly competent stingers. So I would suggest that if you're engaging in collecting these animals, that you have multiple layers and at least a 2 mil wetsuit. Of course, that becomes a problem for hyperthermia. So what I will do is I will bring a bottle of water with me. I don't want to put seawater, but I'll keep putting water and drinking water. So you do have to take care of the body temperature because it's easy to overheat wearing so much protective gear.
Ya, baik, Dr. Budi Utomo, mungkin ada yang ingin disampaikan atau ditanyakan lebih lanjut?
Itu nanti ada pertanyaan kedua ya. Ya,
Oh,
I thank you
ada.
very much for the answer, Professor.
Oh, there's a second question from Dr. Budi Utomo also. Excuse me, Prof. Yanagihara, we usually tell that for the first aid treatment, after removing the tentacles or mucus with a pair or of tweezers, is it effective if we rinse the area with 5% of vinegar? It is easy to be made and very cheap too. Thank you.
I'm sorry, is the first step the tweezers or the first step the vinegar?
The first aid treatment after removing the, maybe Dr. Budi Utomo can explain clearly.
Excuse me, Prof. If we usually tell that for the first aid treatment, after removing the tentacles or mucus on the skin with a pair of tweezers, is it effective if we rinse the area with 5% of vinegar?
I'm sorry, is the 5% vinegar before the tweezers or after?
After the
Okay.
tweezers.
So I think this is a really, really important question, and thank you for asking that. What we find is that any pressure on the tentacle, it's a live tentacle, will cause massive discharge, not just at the place where the tentacle is pinched by the tweezer, but further down on the tentacle. So before applying the tweezer to pull, it's better to flood the area with the vinegar first, because that basically acid fixes the tentacle itself. And you'll see the tentacle kind of start to shrivel a wee bit, but it also inactivates it. So before any kind of physical perturbation of the live tentacle, we find the best thing to do is to flood it with the vinegar first.
Yeah, thank you very much, Professor.
Okay, for the next question from Priscilla Sayogo. Good morning, Prof. Yanagihara. Thank you so much for the presentation. I would like to ask if there are any comorbids or conditions that could make the effect of the venom worse. Thank you.
So all of the effects that I described today are dose dependent. So from that standpoint, your question about comorbidities, what could increase the dose, certainly children have thinner skin than adults, so you wouldn't really call it a comorbidity, but there is definitely an age sensitivity, and women have thinner skin than men. So if you have children or women, and then the part of the body, again, it's not necessarily a comorbidity, but it is a dose-dependent effect. So, for instance, being stung on the neck would give a higher dose than being stung in thicker areas of skin. In terms of authentic comorbidities, basically, you're having an effect in which the kidneys can help clear potassium to a certain degree, but if there's any compromise of kidney function, that seems to be a probable comorbidity. I could think about it a bit more, but just off the top of my head, those are the things that seem logical to me.
Okay, seems clear. Priscilla Sayogo, bagaimana? Ada yang mau ditanyakan? Priscilla Sayogo, ada lagi yang mau ditambahkan untuk didiskusikan? Oke, saya rasa tidak ada ya. Berikutnya ada pertanyaan, and the next question from Dr. Winta. Aloha Angel, the victims of box jellyfish sting might get traditional aid and they might try to scrap and remove the tentacles. Would that approach just make their condition get worse?
So this is the probably the thing that scares me the most. When we did the various experiments in the blood agar, as you saw, the scraping increased the venom load more than 10 times. We did the same experiments with the live piglets, and we found a massive increase in the pathology when we scrape the tentacle. So as far as first aids that should be discouraged, utterly discouraged based upon this evidence, I would say that scraping, it would be the #1 thing to discourage. And it makes sense why the clinician or the first aid caregiver might want to do that, because these tentacles are all over the body and they're very sticky, and they, look very horrific on a child or a victim. So one wants to render aid and remove the offending agent. So again, it's a bit counterintuitive that by scraping or trying to remove the offending agent, in fact, you are increasing the venom load. So again, flooding the site with vinegar is a good way to clear the victim of these sticky tentacles. This Sting No More spray that we've created basically has vinegar plus urea, and the urea really facilitates the removal of the tentacles. So it helps dissolve this sticky mucus type substance that the tentacle also discharges on the surface. So aside from injecting venom into the body, it also exudes this sticky material which causes the tentacle to stick. So either vinegar itself or vinegar amended with urea, and I can share with you the components for this spray, and it's all completely available at no cost as a humanitarian donation in any part of the world in which coastal people are at risk. But again, it makes sense that one would want to remove the tentacle. But again, that's a double-edged sword, and it can massively increase the amount of venom that is injected into the victim.
Okay.
Thank you very much.
Yeah. And then the second question from Dr. Winta. The health worker might see the symptom of box jellyfish look like an anaphylactic effect. What happens if the victim gets the epinephrine injection?
Well, thank you for that question, Dr. Rienta. So this is another area in which the research really surprised us, because in true anaphylaxis, such as in bee sting, the patient has already previously been exposed to the antigen and developed circulating IgE. And when the antigen is represented to the victim, they will have a very high mast cell response, leading to histamine overload and breathing difficulty to the point of even cardiac arrest. And in that case, the epinephrine is a life-saving intervention. In this case, however, the histamine overload is due to a short circuit, if you will, of the system, in that the pore-forming protein itself can cause histamine to be released from the white blood cells. So you have a massive amount of histamine, but it isn't IgE mediated. And you also have the massive release of the catechol from the platelets. So you've already had tachycardia and the hypertensive phase. So by the time the patient presents at clinic with bradycardia and hypotension, the clinician may presume, they hear the word sting, and they may presume that epinephrine would be a life-saving therapeutic. But because the patient has already had basically a very high spike of catechols from the platelets themselves, what's happened is that you've had end organ failure and death due to cerebral hemorrhage. So these patients are not bee sting victims. Box jelly stings are very different, and we have yet to find any IgE-mediated histamine overload. So the clinical course and the biochemical course really are not are contrary to the utility of epinephrine. So there are other ways to manage the histamine overload, and I've had that on that main slide summarizing all the agents we're investigating.
Thank you.
Dr. Winta, bagaimana? Apakah ada yang ingin didiskusikan lagi?
Sudah, terima kasih.
Dr. Winta, tadi yang pertanyaan itu tentang penelitian lanjut apa yang bisa
Oh iya, pertanyaan terakhir Dr. Sasti tentang kolaborasi.
Oke, mungkin bisa sekalian.
Aloha Angel, you have already had extensive research on box jellyfish and Venom. What do you expect from the potential collaboration with Hang Tuah University related to education, research, and community service?
Well, basically, I present myself to you as a willing partner, however you see fit. I think that the coastal communities at risk, for instance, Dr. Munawir introduced me to Mr. Budi, a fisherman in near Situbondo, and these folks have, very interesting lives and life experiences. So I think it is useful to learn how to create communities with coastal stakeholders and to really query them to find out more about traditional knowledge and traditional approaches. For instance, in the Philippines, we found that morning glory was repeatedly suggested. It's also suggested in Thailand. And I did bioassays in both localities using morning glory slurry that I just macerated in a mortar and pestle with saline. And it did, in fact, slow down the hemolysis in the blood agar assay. So I think developing interactive kinds of teams where coastal folks are listened to, as well as presented with data, and to the extent that one can actually do hands-on kinds of demonstrations to show, for instance, with the blood agar, we see that applying the hot pack, you can see with your naked eye over the course of 4 hours that the cold was much worse in massively increased the amount of wounding than the hot pack did. So I think that I'm available to collaborate and be part of this team, but it really needs to be driven by the locality, specific group. And I think for you all as medical educators, you are uniquely positioned to know who your coastal communities are and to kind of reach out, maybe nurses or whomever might be first responders. I think it takes a team, and as a foreigner, obviously I know my lane, so I have just I can provide the data that I have, but I am very excited and open to whatever collaborations make sense. I think we met with a School of Veterinary Science there to work, potentially consider doing a piglet model right there in Surabaya. So I think all of these opportunities, should be continue to be explored. Basically my purpose here today is just to kind of circle up and share the work that I've done and to let you think about ways that seem most logical to go forward.
Yes, thank you. So there are many possibilities for the collaboration, including local outreach program with the fishermen and the community, and then we can try traditional herbs or etc. For the first aid, and we can have a research on piglets with the veterinary faculty of Airlangga University that you saw last time. Okay, thank you.
Terima kasih, Dr. Winta. Okay, and the next question from Calvin Husohandono. Hello, Prof. Yanagihara. Would the use of adrenaline be a positive intervention in case of hyperkalemia due to the RBC perforation?
Again, that I alluded to that earlier with administration of adrenaline or epinephrine. So adrenaline and epinephrine are synonymous and unfortunately have led to death. So using EpiPen has led to this, what's called end organ failure. So after having already had a hypertensive event, especially the blood vessels in the brain are uniquely vulnerable to a second bolus and a hypertensive event, such that one could have end organ failure. In fact, that's what's marked most of the deaths from Iroconji syndrome have been well-intentioned, but probably completely inappropriate use of epinephrine and leading to cerebral hemorrhage. So again, adrenaline would be an absolute no-go. And from the standpoint of our work in piglets, we have other mechanisms to manage the histamine overload. Hyperkalemia can be managed through either supporting kidney function or doing various other mechanisms to reduce the plasma potassium, but administration of adrenaline itself isn't a clinical management for hyperkalemia.
Oke, thank you, Prof. Bagaimana Calvin Yusuf Handono? Apakah ada yang ingin ditanyakan lebih lanjut perihal hiperkalemi?
No, thank you, Prof. Yanagihara. It seems as if Dr. Winta and we asked this exact same question. So
That's okay. Great minds think alike.
Okay, another question from Adi Husodo. Good morning, Prof. Yanagihara. It was a very insightful presentation. Is there any gaps that you found in this research that could be fulfilled by further research in the near future? Thank you in advance.
So, yes, obviously I've spent 24 years to get to this point, but in that one slide where I showed all the potential pharmacologic pharmaceutical interventions that we're investigating right now, that's another 20 years of work. So we do have ongoing critical unmet need areas, if you will, and that the main gap right now is, okay, this technology could stop the sting if administered quickly enough. But if your patient doesn't arrive to the hospital until 2 hours after the sting, and they have all of these things going on, histamine overload, they have hyperkalemia, they have all these things going on, what is the best modality to manage them at that point? So that's what we are doing systematically presently.
Gandang hapon.
Okay, Adi Husodo.
Ayun lang daw.
Thank you, Professor Yanagihara, for the answer. Thank you.
Okay. Oh. And then the next question from Ayu.
Matindi.
Good morning, Prof. Yanagihara. Can a dead still sting us? In case we found or step on one of them on the coast or sand accidentally? Thank you.
Dina.
Halo, tolong yang lain di mute ya. Tolong yang lain di mute itu.
Sir Noel, I think you're not mute.
Continue, please.
Okay.
Continue.
Yes, so they aren't dead in this case. The tentacles, well, it's rather surprising. One can lyophilize these Nidae and they can still discharge. I had some sample given to me that was a 30-year-old freeze-dried sample of Nidae from Chironex from Australia. And I was wearing double gloves and doing an experiment, and somehow I got one Nidae on my skin and it discharged and it left a huge black wound. So here it is 30 years later, it's not dead. So these Nidae are amazing storage capsules that live beyond the life of the tentacle itself or the organism itself. So stepping on them on the beach is not safe.
Okay. Thank you, Prof. Bagaimana Ayu? Sudah cukup menjawab ya tentang apa yang ditanyakan? Ayu? Oke. Okay. Apakah masih ada pertanyaan dari para audiens kalian di sini? Silahkan bisa raise your hand atau bisa tulis di chat. And actually, I have some question for Prof. Yanagihara. there is something that caught my attention from your presentation. It is about cytokine storm in Iroconji syndrome, which is in my question is the cytokine storm that occurs in Iroconji syndrome same as the cytokine storm when the human body experiences an infection? Because all I know about the cytokine storm is when the cytokines that rise immune activity become too abundant, the immune system may not be able to stop itself. So immune cells spread beyond infected body parts and then gobbling up red and white blood cell and also damaging our organ. So is that what will happen? Is that the same?
So that's a great question, and that is ongoing work. Basically, what we've done with our piglet model is we've been able to get a dose of the live tentacles that leads to Iroconji syndrome. So they have pulmonary edema, they have all of the hallmarks of Iroconji syndrome, and we've taken time series of the blood to do the cytokine levels. Unfortunately, for the clinical human cases of Iroconji syndrome, you might imagine that we don't have cytokine panels that have been systematically done. In the field, when Iroconji case presents, the clinician is all about managing them. But comparing the piglet dose response from the live tentacles with piglet models that we have already from sepsis, we see many of the same cytokines elevated. So I can't answer your question completely presently, but in a general sense, yes, there are similar cytokines being elevated from the Iroconji syndrome that's based upon the venom exposure as there are from the bacterial sepsis.
Thank you, Prof. Mungkin ada pertanyaan lain dari para audiens di sini. Kami persilahkan, mumpung masih ada waktu yang tersisa. Or maybe another question from me, actually. All the mechanism that occurred, what is the most likely to cause a person to die after stung by this jellyfish?
Well, so again, it's dose dependent, but the most likely cause of death acutely is this hyperkalemia. Was that your question?
Okay, hyperkalemia.
Yeah,
hyperkalemia. Yes, the pouring, the effect of the pouring on our RBC, right?
Well, right.
Okay. Thank you, Prof. Baik, apakah masih ada pertanyaan lainnya? Oh, ini ada pertanyaan from Dr. Faridianto Yudo. What is more destructive to RBC, the jellyfish toxin or bacterial toxin? Streptolysin, streptokinase, and what type of hemolysis happen? Total hemolysis or partial hemolysis? Okay.
So we have done the HU50, so the dose of the hemolysin at which 50% of the red blood cells are lysed, and that's about 7 nanograms. So for anthralysin O, which is the most potent of the bacterial lysins, it's about 20 nanograms. So streptolysin is less potent than anthralysin O. But the chirodropid porin is even more potent than that. So to answer your question, then the most destructive to the red blood cell are the chirodropid porins. As far as what type of hemolysis happens, what you saw in the microscopy time series, you have crenellation, which is before, hemolysis is generally defined as the loss of hemoglobin. So hemoglobin can only exit these pores once the red blood cell swells. So they already have the pores functionally inserted into the red blood cell membrane, and there's already loss of potassium. So one wouldn't necessarily call that partial hemolysis, but that is that you have now created holes perforating the red blood cells, and you see the swelling of the red blood cells. But again, if I remind you of that microscopy time series, they had to swell quite a bit before they blinked clear. And that moment at which the hemoglobin leaves the red blood cell, it then becomes a red blood cell ghost. So that would be what you might call hemolysis. But prior to that, the loss of potassium can already be catastrophic in and of itself. So folks in the field have said, well, this corn can't possibly be the explanation for death because we have folks who have died and their plasma hemoglobin is not elevated. Well, again, from the microscopy, we see that the plasma hemoglobin isn't the first thing to leave the red blood cell. And we have published on this with ion-specific electrodes in the plasma, and one can measure the release of the potassium from the red blood cell, and that happens within seconds. So whereas complete, if you will, hemolysis and loss of hemoglobin takes 8 minutes, the loss of potassium happens very quickly. So I hope that addresses your question.
Yeah, thank you, Prof. Yana Gihara. I think it's a completely total destruction of the red blood cell because the porrin will make the red blood cell leaks and it will lose the calcium from the cell and it will affect the cardiovascular system, right? Thank you again for your explanation.
Thank you. Apakah ada pertanyaan lainnya?
Ada.
Ya, silakan Bu Dekan.
Nagihara, excuse me. How frequent do you see the box jellyfish and maybe other friends or divers are more frequent, or I think as a divers, I do not yet know. I do not yet see the box jellyfish yet. Yeah, but this presentation is very good, very nice for us to see how box jellyfish as a killer, yeah, as a killer. But not frequently we will see the box jellyfish. Maybe Prof. Gihara have an experience many times to see the box jellyfish or not? Thank you.
So that's a good question. It's very difficult, in fact, to work out the exact locality and the time in which these animals are found. So they are not constitutive members of the coastal area at all times of the year and in all areas. They have very specific habitats. So that's good news from one standpoint in that we can use that information to really provide proper warning to coastal communities about where and when these animals are existing. But yes, they're not in all places at all times. And folks I've done field work will be able to share with you that we work pretty hard to find them. So that's good news.
Okay, thank you very much, Professor Jana Gihara.
Terima kasih.
So don't be afraid, yeah, Professor, yeah. So don't be afraid as a diver, as a community, yeah.
Yeah.
But we should be very careful if we meet or we sting of the box jellyfish. Okay, thank you so much.
Baik, sepertinya pertanyaan dari Bu Dekan adalah pertanyaan terakhir menutup sesi tanya jawab kita. This is the end of question and answer session. Thanks to the audience. Yeah. Excuse me.
Oh enggak, sepertinya ada pertanyaan dari Indah Afifatul Husna, DM.
Indah Afifatul Husna.
Dibacakan lagi.
Yeah.
Good morning, Prof. Good to see you again in these opportunities. Okay. As we know, and as the paper that you give to me before, phenobarbital can affect to our red blood cell and causing iron efflux immediately and caused cardiovascular effect, and how much time that needed till this iron efflux caused a death based on the research or the accident that you may know. And then so we can know the golden period to give the first aid for these cases, because as we know, some of coastal areas is far away from the healthcare facilities.
I had a little trouble understanding the question.
I'm sorry, maybe I can type it in the chat column, right? But I have a bad signal here.
I have put in the chat the link for the lovely talk that Dr. Munawir is giving in Indonesian language, so the students can listen. I'm sorry for the technical difficulty from my computer. I couldn't share it with you, but do you see it in the chat?
Yes, we can see it and we can download it. Thank you.
I've shared my question in the chat box.
Yes, okay. This is question from Indah Hafifatul Husna. Good morning, Prof. Good to see you again in this opportunity. As we know, Venom can affect our red blood cell and cause K plus efflux immediately and cause cardiovascular effect. How much time that need till this K efflux cause a death based on the research or the accident? So we can know the golden period to give the first aid for this case. Because as we know, some of coastal area is far away from healthcare facilities.
May I ask, Sir Noel, are you online?
Yeah. Hello?
Sir Noel?
Pardon?
Yes.
Oh, yes. So I want to share the answer with Sir Noel, who actually made the introduction in Tagalog Barangay, and we met Nurse Heidi. And so we presented this data that the child could present and look dead, but please do rescue CPR for up to 45 minutes with the rescue breath. And so about 9 months after we went to that locality and gave that talk, little boys were playing in that same area where I showed the video, and one of the children was stung, and his friends pulled him out, and he was dead. The tongue was out of his mouth, no heartbeat, no breathing, and the children cried to the area around and ran to get Nurse Heidi. So Nurse Heidi remembered our visit and had them go get the Sting No More spray, and she commenced to do the CPR. So the child had not been breathing or having a heartbeat for 10 to 20 minutes. We're not exactly sure. But the rescue CPR with the rescue breaths revived the child within 15 minutes. And the boy is now alive. So Nurse Heidi was recommended for the Heroic Nurse of the Year of the Philippines. Dr. Sagio, can you mention about that?
Yes, thank you very much, Dr. Yanagihara. To my fellow researchers and professionals, good afternoon. So yes, one of the engagements that Dr. Yanagihara had in the Philippines was really the translation of educating coastal communities in life-saving techniques. So what he did was gave us a pretty good lecture, and somehow some of those lectures were translated even in the vernacular in the Visayan region. The area that she mentioned is in that's near Kaluwayan Beach. It's in Samar. It's in Marabut, Samar, Central Visayas. So the thing is, just like probably, I posted a question here regarding coastal community education. Probably with regards to our engagement with life-saving techniques in coastal regions, one of the effective ways that Dr. Yanagihara did was to educate that community. She was so specific in her training programs that one of our nurses here, she's a barangay health worker. We call it a local health worker, but she is a licensed nursing. She's a licensed nurse. Then she remembered the trainings that we gave, coupled with the fact that she's also a nurse, translated into saving a boy that was stung near a very much it's a well-infested area with jellyfishes. So when she was awarded with that Hero of the Year award for nurses, we suddenly remembered that these kinds of trainings so that it can translate to life-saving a program, right? So I hope, right? I hope further training and further involvement and probably being transparent in applying all the technologies and the trainings will eventually translate into lesser deaths, or probably we call it here unwanted deaths, right? Because you simply do not want a person going outside in the beach and then suddenly you have to go home with a dead body, right? So that is how we want to translate these kinds of program. And with regards to the training program, I would say that the program that we left in Tagalog is sufficient enough to say that it's working. Okay, it works. The technology of Dr. Yanagihara plus the involvement of coastal communities really translates into evidently positive outlooks.
Ya, mungkin Dr. Sasti, pertanyaannya tadi kan berapa lama ya? Karena kita sering jauh dari because we berapa lama?
Excuse me, Angel. Actually, Indah is asking you about the golden period to save life.
So I think that's a difficult question to make an absolute answer on, because the course of the individual victim is dependent on the dose. So at a certain dose, you have death within minutes. And so if it's a child and you can have the CPR with rescue breaths, children have more of a golden period than adult, but at a certain point, no amount of resuscitation will be useful. So I don't know that anybody would be able to answer this question precisely. Most of the stings in adults, however, have serious necrotic wounding, a lot of pain, and they may have this kind of hypotensive presentation at clinic. So if they've been stung 2 hours ago, they probably the amount of venom that was injected was insufficient to cause death, but they still will have this course that comprises Iroconji Syndrome. So with regard to that golden period, that's more of a critical care period that has to go out basically to 48 hours of watching them and being able to offer supportive care, oxygen, etc. So I think this is a sufficiently complex kind of situation where one can't give a direct answer. I know the question is a very important one, but I'm sorry, I don't see that we have enough evidence to give a conclusive, absolute answer.
Thank you, Prof, for the answer.
As soon as possible, maybe.
Thank you, Doctor.
Time left is 45 minutes, yes, Prof, for stable until they realize that they should get some Iroconji syndrome, right?
So, for instance, in my case, I had Iroconji syndrome, and I was offshore, so I had to swim to the beach, and I'm not even conscious of the last part of my swim. I was in such agony. My throat was like on fire, my arms, my lungs were filled with fluid, and I was wheezing. I was just kind of an automatic pilot. But when I got to the beach, I passed out, and then I came to consciousness in an ambulance. So, you know, this kind of from that point until actually 72 hours later, I was in agony. So, but I had no proper care whatsoever. So I think the golden period to give first aid and care, it's dose dependent, it's victim dependent, it should be directed by the signs and symptoms. But another questioner asked earlier, what are the gap areas? I think this is a very good one that Inda has pointed out. This is a good gap area to create standards of care that are driven, that can sort of answer this question about the golden period. But basically the bottom line answer is as soon as possible.
Okay, thank you, Prof. Is there any question more from participant in here? Apakah masih ada pertanyaan dari para partisipan di sini?
Sudah mungkin Dr. Sasti.
Para dosen barangkali ada? Para dosen
mungkin ada yang mau ditanyakan?
Ketua, Dr. Westi atau mungkin yang lainnya mau ingin bertanya? Halo.
Atau mungkin nanti ada sesi 5 menit setelah audiens keluar, Bu Dekan, untuk membicarakan.
Ya, mungkin setelah Prof. Yana Gihara, sebelum mahasiswa keluar ya, mungkin setelah Prof. Yana Gihara leave, saya ingin menyampaikan sesuatu untuk khususnya mahasiswa, ya, karena menyangkut visi misi kita. Oke. Kalau tidak ada nanti.
Baik, Bu Dekan.
Baik, dengan tidak ada lagi pertanyaan yang kami dapatkan di sini.
Tapi jangan keluar dulu semua ya, jangan dulu keluar semua. Barangkali Prof aja.
I would like to say thanks to Prof. Yana Gihara for the information and knowledge which have been given to us. This is very useful for us. Lalu acara berikutnya adalah pemberian sertifikat kepada pembicara, kepada Bapak Rektor Universitas Hang Tuah, Profesor Dr. Insinyur Supartono. Kami persilahkan secara simbolis menyerahkan kepada Prof. Yana Gihara. IT mungkin bisa di-share screen untuk sertifikatnya.
Itu ada di belakangnya Dr. Sasti. Share screen.
Oke, silahkan kepada Bapak Rektor.
Monggo Bapak Rektor berkenan menyerahkan sertifikatnya.
Yes, okay. Thank you very much for Angel Ani Yana Gihara PhD. This is certificate for you, and I can to speak. Thank you very much for you speaker now. Thank you.
Terima kasih Bapak Rektor.
Ya.
Dari Prof. Yana Gihara menyampaikan sesuatu.
Silahkan Prof. Yana, ya. Prof. Yana Gihara, maybe you want to say something. Thank you.
Well, thank you very much. It's been a great pleasure. And even with our strange world of COVID, I feel somehow we're all together.
Yeah. Thank you very much.
And the collaboration will develop, yeah. The collaboration will continue, and maybe from our rector will develop to maybe other faculty in your university, collaborate with Hang Tuah University in the next future.
I look forward to that very much. And I'm really impressed by the students these days with the resilience. So I want to give a special thank you to the medical students who are working on their education, and they are being very flexible and resilient to have all kinds of new learning methods. So I'm very happy to be with you by Zoom. And I look forward to when we can work together in person. But I think this is a great opportunity to, learn how to learn new things and to really reach out. So I'm very happy to have our Philippine colleagues also online in this event. So this is really kind of a remarkable opportunity to think about an interesting topic and how we can move forward together.
Yes, thank you very much.
Thank you very much, Prof. Baik, akhirnya sampailah kita di penghujung acara pada siang hari ini. Kami ucapkan terima kasih atas waktunya dan juga partisipasi para partisipan sekalian. Wassalamualaikum warahmatullahi wabarakatuh. Salam sejahtera bagi kita semuanya. Om Santi 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 Namo Buddhaya. Salam kebajikan. As the master of ceremony, I would like to thanks to Professor Yanagihara. May what we had conduct will be useful for us. Thank you.
Thank you, Prof. Yanagihara. Thank you, Dr. Sahwil.
Dr. Sakti. Nah, setelah Prof keluar, baru kita bicara, ya. Mohon izin Bapak Rektor.
Iya.
Bapak Rektor.
Ya, saya keluar dulu kalau gitu, biar tidak.
Terima kasih, Bapak Rektor.
Bapak Rektor nggak apa-apa, Bapak Rektor kalau mau dengarkan.
Kebetulan ini mau sholat dulu.
Oh ya, silakan Prof. Ya, silakan.
Ya, oke.
Terima kasih Pak Rektor dan para warek.
Terima kasih, Prof.
Jadi, terima kasih.
Terima kasih. Ya, izin lift ya, Bu.
Iya, terima kasih Bapak warek.
Terima kasih.
Ya. Jadi untuk mahasiswa, mohon maaf untuk mahasiswa, karena ini terkait sama visi misi kita ya. Bagus sekali bahwa yang disampaikan sama Prof. Yanagihara ini akan sangat berarti ya untuk kita sebagai negara bahari ya, negeri kepulauan ini melakukan penelitian-penelitian yang masih lebih banyak. Ya. Jadi berbahagialah karena visi misinya UHT, khususnya juga FKUHT ini adalah dengan kelautan. Tapi saya hanya ingin sampaikan bahwa yang disampaikan Prof. Yanagihara itu bagus sekali untuk kita ketahui. Maka tadi pertanyaan saya adalah jangan terlalu khawatir ya. Nanti kalau gitu saya takut